So today I'd like to introduce our first speaker, Dr. Tomas Kalina. He's an associate professor at the Department of Hematology and Oncology at Charles University. And his talk title is Standardization of Cytometry and Antibody Validation. Tomas?
So, good morning. I hope you can hear me well and you can see my slide now.
Yes, we can.
Great. So my name is Tomas Kalina. I am working in Prague at a Child Leukemia Investigation Prague group. I've also played some role as a counselor in ISAC, and I'm leading seminars and courses on flow cytometry. And today I would like to share with you a talk on standardization of cytometry and antibody validation. Why standardization? So standardization kind of was something that I never seek, but it kind of thought me, and it found me when I was joining a Euroflow group, where I was responsible for the standardization of hematological malignancies and later primary immunodeficiencies diagnostics. And why antibody validation and how this is connected to standardization. This found me when I purchased Cytof into our institution, and I realized we need to conjugate antibodies. And I realized how difficult it is to find a good antibody and how difficult it is to realize whether we use a correct antibody or not, if we succeeded in conjugation or not. So through this, I learned quite a few things that I want to share with you today. Sorry, I'm still getting to understand my computer. So what do we report from cytometry? So that's an important question, because cytometry is not only one, but there is many different topics and assays that have a different aims. So often the clinical cytometry reports numbers. So it's actually doing enumeration in CD4 counting, CD34 counting, or minimal residual disease evaluation. It also can report immunophenotype as some sort of a characterization of cells of interest. This is especially true for leukemia diagnosis, where we need to assess which type of leukemia do we diagnose at the moment, and share this information for the benefit of the patient. And we can actually go as long as to analyze and report complex patterns. So for instance, we want to make sure that what we see is a normal B-cell development and it's not somehow altered due to a disease or treatment. Or we can report patterns of phosphorylation when we assess multiple phosphorylation sites at different kinases at once. So what do we actually need to measure reproducibly? So there are competing features of these cytometry assays that I want to highlight here, so that we understand that not every assay has the same needs and same requirements. So what I will be talking about today is mostly the third issue, which is kind of the relatively low complexity assays, clinical, that are 8 to 10 colors, and we need to perform the same assay over and over for different patients at different time points. And in fact, our experiment is probably a year-long measurements of different patients or even more. So we desperately need to use some sort of standardization so that we can compare what we measure today to what we have measured half a year ago, and to those samples that will come to our lab in 3 more months from now. We want assays that are easy to use and easy to measure reproducibly. We want to get done fast, and if possible, we appreciate automated analysis of those assays. So the term standardization that I use here is a set of assays or set of methodological steps supported by standard operating protocols. And here we aim to remove any sort of variation that may come into that assay. So some other groups are using a term of harmonization, or they use the same term standardization, but with a different context. So here we try to fix everything as much as possible and use it over and over in our and other labs as well. So the different cornerstones of standardization is the instrument setup, where we use calibration beads such as rainbows. And essentially, we prefer an approach that one setup fits all of our assays. We then standardize the sample preparation, which is mostly the cell preparation plus staining, where we use panels of reagents that are defined. And if there are any alternatives in the individual reagents, we carefully test them and compare to each other. And then we also have some methods how to use computational tools to help us in our analysis. So the beads, you see here rainbow beads that I always suggest that anybody should be using those beads to first check the performance of his or her cytometer, ideally every day or at the cold start of the instrument. And this helps us to trust the cytometer and realize whether or not there are any issues happening that particular day. But the rainbows are not spectrally matched. So you will be good using rainbow beads as a beads to achieve a standardized setup on a single machine or in the same type of a machine, but there will be some issues when you want to transfer those measurements to a different machines with a different spectral properties. I will talk about it later. The SOPs or standard operating protocols that we use are an SOP for instrument setup and compensation that we set up for Europe, our members and followers. There is also a separate standard operating protocol for sample preparation and staining. And we use defined marker sets that we would call panels. And these are using an antibody that we call reference antibodies, so those that we used for the first time. And because this is going on for many years now, actually more than a decade, we have also developed and tested a catalog of alternative antibodies that perform exactly the same as the reference antibodies. They might be the same clones from a different company. There might be a second generation of the same clone from same company, but they also might be a different clones from a different company that perform exactly the same. So the result of standardization can be seen here. This is a concatenate of 27 samples that were measured on a healthy donor's peripheral blood in 9 different labs. And you can see that the light blue line is a CD4 on a CD4 T cell. So we achieve pretty good standardization on CD4. The CD20 on B cells is still relatively good, but not as perfect as the T cells. And what is the most difficult to standardize is actually the negative signal here in the NK cells and the CD8 T cells. And the difference you see between the different samples is actually caused by improper compensation by in the different instruments in different labs. Nevertheless, since the in cytometry measure in a logarithmic scale, what really matters is the difference at the high end of a scale. That is, if you use this all information from all channels and you run a principal component analysis, you would realize that actually all the different subsets cluster together at particular spots of the PCA. So we can reconstruct the same phenotype of the critical subsets that are gated in this tube across all the labs, and we can analyze them together. This was done for the purpose of a quality assessment of the patterns that I will mention later on. So at what level and what extent of reproducibility do we need for different different cornerstones of the whole cytometry assay? So if we aim for instrument setup as a standardized part of the procedure, our opportunity is to gain a perfectly same data pattern. So the be exactly the same. The challenge we will realize is when we need to upgrade to a new cytometer, or somebody from the same consortium buys a different cytometer from different vendors. So we have to somehow handle the differences in the hardware. We can do a common marker choice that will allow us to reach much better and much higher reproducibility in our results. However, we will be challenged by the need to make a consensus and or perform some benchmarking if there is no consensus on particular markers, but we want to make sure which one has the better marker for that given purpose. For sample preparation, the opportunity is that, again, we will get reproducible measurements. However, we will be challenged by the different habits in different labs that are very, very strong opponent. I never realized that maybe 10 years ago, as much as I realize it now. We will be challenged by the SOPs that are already in place and need to be changed, so people don't want to do that. And obviously, there will be some unique needs of particular assays or markers or targets that we need to consider. So it's not easy to find a single sample prep method that will serve every possible assay with the same quality. The assembly of reagents into the panels will help us to achieve a perfectly reproducible assays. However, it's complex, and we will need to do some sort of benchmarking to convince people, ourselves, and the other collaborators that that particular panel that we assembled together is either the same or actually better than the current panel they are using so far. In data analysis, there is a huge opportunity for improvement and for gaining reproducibility and also efficiency through different automated, semi-automated, or even manual steps that will be standardized. And if you want to do that, the ideal of a setup is when the panels or even the whole patterns are the same. It's much easier to develop a computational approach to data analysis that will be comparable. And data interpretation, what I mean here is that if we would be able to call the same cell types the same name, we would really make the communication easier. But again, we need to find consensus. So this is an overview of different consortia, how they employed different parts of this procedure. So some of them really stopped short of maybe even trying to standardize the instrument setup, claiming that this is not possible or not feasible or people don't want to do it. So each consortium took a slightly different angle and prepared a little different sets of rules that either led to a full standardization or to partial standardization or harmonization, perhaps. This is a busy table that I just want to refer you to the paper that was published this year in Cytometry A, where I go into more detail of these assays. And in essence, cytometry, especially in a clinical practice, needs to achieve some parameters of quality, which are called trueness, precision, and accuracy. And again, the key point is, what is the question that we pose? So is it enumeration? That's easy, and we are used to work with those numbers. When it comes to enumeration, it's very similar in biochemistry or in other disciplines of laboratory medicine. However, when it comes to patterns, this is much more difficult, and we will not easily employ all these principles and give them some measurable outcomes. I think that one key question is kind of all the different shades of gray. So how good is good enough? So we all strive to be perfect, but in reality, we are rarely perfect. So the essential question is, how good is good enough? And I think it really depends on the purpose. So in the enumeration, there is consensus that CV of less than 10% should be achieved, except for the assays that track the very low abundance populations where even 20% CV can be acceptable. In the intensity of staining, there is very little data on how good we can get. And we have done this in the scope of Euroflow quality assessment, where we were looking at the MFI of different markers that are stably expressed on a cell surface of leukocytes. And what we measured was that we can get a CV of less than 11% for the stable molecules. And we mostly get below 30% of CV for the alternative reagent benchmarking. So it's still relatively good, but the variability by the target epitopes and target molecules, but also by the reagents. So these two play together, and we have to realize which is contributing more to the spread of the measured data. Is it the biology or is it the reagents that we use? If we want to reproduce data from different projects, we probably need to find a common language on the different definitions of cells of interest. So in leukemia lymphoma, typically there are consensus papers that are trying to achieve just that with variable success. Typically, finally, with the consensus papers, nobody is completely happy. Nobody is completely opposing those consensus papers. So that's definition of a compromise, right? In immunology, it's more challenging because there is not so much of an interest in finding a common language. However, there are exceptions. For instance, the Human Immunology Project Consortium has proposed some definitions and way of identification of different subsets of human immune system. There are partial solutions in terms of OMIPS, which is a special format of publication, Cytometry A, that describes the optimized multicolor immunophenotyping panel. So you can take some inspiration there of how cells are gated, which markers are used for definitions. And of course, the literature is the major source, but it's probably too great to be embraced easily. And the key issue here is what is the correspondence between papers and how do we generate recommendations for a good praxis? Again, there is nothing better than validation and perhaps benchmarking, which is rarely done, but I want to encourage everybody to actually take that step and if possible, perform a benchmark test of your new panel versus the other panel. This is, by the way, a requirement in the newly submitted OMIPS. How reproducibility can be achieved between projects. So this is an example of CD4 T cells, and there is about 4 different definitions of the different of the same cells, naive CD4 T cells. So they can be defined by either CCR7 together with CD45RO isoform, or they can be defined also with CD27 and 45. Also be defined with 27 and CCR7 in some instances. However, you can see that in the 3 pictures, you get exactly the same number. So for the healthy individual, those 3 definitions are the same, and we can benchmark it this way. If we move to CD8 T cells, it's already more difficult because there will be some cells in the CD27 positive region that will in fact be not naive, but rather the TEMRA revertans. So what is valid for CD4 T cell compartment is not the same true for the CD8 T cell compartment. Also, there might be differences when you stumble into the disease sample. So particular diseases can change the normal expression patterns and can be and can lead to some additional sources of variability in the phenotype. we want to also reproduce the measurements between different instruments. So it can be because we want to measure different patients in one lab across larger period of time, perhaps from one instrument to a new one that we newly buy to the service in the lab, or we can compare different patients in several labs. So this is a picture of a easy two-color plot of CD3 and CD19 T cells and B cells. And in fact, this picture is composed of many different measurements from different labs of the Euroflow. So we can achieve the same picture even in the different labs and even using different instruments. So we have done so for a BD-Fax Canto, which was our reference instrument, and then later on we matched the patterns measured on BD-Fax Lyric, Beckman Coulter Navios, and in principle also for Max Quant instrument. There are two papers that I would refer you to. In fact, if we use any instrument that has a similar laser excitation by violet, blue, and red lasers, and similar detectors for at least 8 or possibly even 10 different fluorochromes, we will be able to use the same panel to measure the sample. However, there will be some differences. So one difference is relatively easy. This is a difference that comes range that will be different on a different machines. They use either 18-bit or 20-bit or now even more resolution. So the numbers will be different and will need to be rescaled if you want to compare the samples together bioinformatically or in a same software. And then there will be a differences in the emission optics. So the major difference that we encountered was for the second violet parameter, which was either Pacific Orange, Horizon V500 or OC515 dyes, where some instruments have relatively shorter wavelengths that they detect and some have relatively higher. And if you look at it more, what you see here is a spectrum measured on a Sony spectral cytometer out of the rainbow particles. And the position of the Canto filter is depicted in red and the Navios filter is in blue. So you see that from the same beads, you have a higher level of fluorescence measured in the Canto filter. However, if you look at the 3 different alternative fluorochromes that we have used on Canto, you realize that they are all slightly different. So on Pacific Orange, you will have a maximum emission in the Navios filter and not so much in the Canto filter. Horizon V500 will get the maximum in the Canto filter, but will be lower in the Navios filter. So what that means is that if you ever want to make a standardized assay that would be acquired on 2 different instruments with slightly different emission or detection optics, you will have to adjust the target values for the rainbow beads so that eventually not the beat intensity matches each other, but the measured signal matches the other. And to make it even more complicated, you will have to always make a target value that is specific for a pair of a filter and fluorochrome. So there will be one value for BC Navios and Pacific Orange, and confusingly a different value for the same cytometer, but different fluorochrome, for instance, the Horizon V500. So there the complexity is greater. So every secondary instrument needs specific target values for each distinct fluorochrome. And this is a challenge because in our hands, even the Euroflow panels that are both the published and those in development are using multiple fluorochromes, multiple reagents from multiple companies, and the different companies make their alternatives of fluorophores that are not always spectrally matching. So some of them are numbers of reagents are really huge, and you have to make sure that for each of them, you are still working at the standardized conditions at different instruments. And this is even more for the compensation, from the compensation perspective. So in general, the instruments with similar excitation and collection optics can measure the same panels. It's relatively easy for the low complexity assays than for the high complexity assays to transfer them. And what is really different with different instruments is the spreading error that can be very different in different instruments with a different detector efficiency and different noise. So compensation tubes must always be used that are particular to the reagents and instruments. And now we move to a slightly different topic, and that is the antibody validation and reproducibility of measurements that are coming from the antibodies. So specificity is a term that specifies whether the intended target of that antibody is in fact recognized. Epitope nature is a characteristics of the protein, typically protein that we detect, and in some it can be changed by different methods of sample prep. The cross-reactivity is the issue where also unintended targets are recognized, and antibody dilution, is sometimes called titration, is a key for the right interpretation of the result. Reproducibility and clonal identity means that we can achieve the same immunophenotypic pattern and with the different clones or different batches of the same clones. And this is a problem sometimes that is on both sides of this collaboration. On our side of the researchers or diagnostic flow cytometrists, that we don't pay attention to the details, and sometimes there is a lack of commitment in terms of doing the titrations, doing the validation. And on the other hand, there is also some role for the companies that they should provide the best reagents and the best characterization for those reagents. These issues are summarized of this year. So antibodies as reagents. So we probably all know the seed nomenclature, even though not everybody is really busy thinking about it anymore because we take it as granted. This was originally and still is run by HLDA workshops where new clones for new targets are assigned a CD, and these panels of antibodies are evaluated by a panel of experts by wet experiments. The HCDM is the organization that runs those workshops. It also runs the website where each antibody that was ever part of a workshop is mentioned, so we can find which antibodies were tested in particular CD workshop, or against which CDs we have available reagents. And this is a recent paper that is summarizing the different nomenclature and achievements of the HLDA over the years. So what is important for each of us? So I think very important and often overlooked is the fact that a clone name is some sort of identity of the reagent, of the clone. This is important because it is linking that particular clone with its history, with its published history. So one important question is, was it workshopped ever? And if not, is this clone something that can be used for tracking the performance in different studies in the literature? Specificity means that we know what is the target of that antibody. In the old days, it was typically evaluated in the HLDA workshops. Nowadays, it can be already engineered by using a particular immunogen or epitope, or established by cross-blocking experiments with other antibodies. We also want to know whether the immunogen was native or denatured, because it then has an important information about which type of assay can be used to do the sample preparation. So if the sample preparation will be using fixation, we will better use an antibody that was developed against denatured immunogen. If it will, if a native used, we have to test whether fixation alters this or not. Reactivity, so we typically seek reagents that are reactive against human targets, but many can be cross-reactive with other species. And application, so we typically seek reagents that were tested for flow cytometry. Oftentimes, if we do a surface staining, immunoprecipitation under native conditions is also a method that typically gives cross-reactive antibodies between, that work for both cytometry and IP. However, Western blot tested antibodies that were used under denatured conditions are typically not good for flow cytometry under native conditions. Again, the difference is when you try to do a nuclear staining where you do fixation as one of the initial steps of sample prep. So then these epitopes are also fixed, so you will get an antibodies that perform well for Western blot and also for flow cytometry. For validation, so now if we either conjugate an antibody for Cytov or we receive a new reagent, we would like to know whether there is a reference clone that should perform in a known way, and whether we can compare our newly conjugated antibody to this reference antibody. If we are on our own without any reference clone, we want to know at least what are the cells that are positive for this antibody, and also importantly, what are the known negative cells. So we want to make sure that the positives are really positives and the negatives are negatives. And again, we want to double check whether the application is listed and we use it properly. So this is a summary picture on the antibody validation, where in the first panel, there is all the antibody information that we have just talked about as an example of a complete information on antibody. We can also see a validation performed on a transfected cell line. So this is probably what the manufacturers might do, or people that still develop antibodies in order to ensure that they achieve the right antibody, they raise the right antibody when they are developing new ones. Then it's a good practice to test the known positive cells. Here it's a Raji cell line in blue, and the known negative cell line, here it is K562 in green. And you can test this also on a primary cells like human lymphocytes versus monocytes for this marker. And you can also compare it to the reference clone that can be used for comparison. Furthermore, IP or Western blot can be done to ensure there is no more than just a single band and single reactivity, again, depending on the type of antigen or epitope that you are detecting. You have to make sure that you are using the right either native or denatured conditions. This is a picture that shows a performance of 4 different clones that are all targeting CD4. So you can see that, I'm sorry, this is targeting CD3. And you can see that the 3 of them give you exactly the same histogram. So they are perfectly comparable. And the third one is slightly lower. And this is a constant feature of these 4 clones. So again, you can use flow cytometry for benchmarking of clones. And in the ideal situation where the conjugation that is performed is 1-to-1, for instance, this is true for phycoerythrin, not for some other small molecular dyes, you should at the end receive the same pattern if you have the same similar performing clones against the same targets. If you don't know what to expect, you can consult this resource on our HCDM website, CDMaps, which we have developed recently, and where you can find how positive or negative are different markers at different subsets. So here I choose CD4 T cells, naive CD4 T cells, and I show that they are negative for CD19 and CD8. They are positive for CD4 and for CD27, and you even can read out quantitative information. And this resource was published in Frontiers in Immunology last year, and you can select any be measured and any number of monoclonal antibodies. The problem of performance and stability is actually really an open issue where not much data is available. The vendors are always claiming that they do quality control and they do it well, but the parameters of the quality assessment or quality control are typically not shared with the users. And I think this is one of the points where we should probably negotiate with the vendors, that we want to know whether a given reagent is really a stable one, or it can differ very much. And it can be a biology. There doesn't have to be same criteria for any different reagents, but we should probably have some benchmarking. And the better stability and performance, or better stability, is achieved mostly with the dried reagent. So this is probably a future of the clinical and standardized cytometry, to use the dried mixtures. And finally, this is my last topic. If we know that we can achieve a particular median fluorescence intensity, we can achieve the reproducible staining in a different donors, in different labs, in different instruments, and we can use it for quality assessment. So this is exactly how we developed quality assessment rounds for the Euroflow protocols, where we can run samples either on Canto or on other instruments, and we can achieve the same median fluorescence intensity. And if our measurements are either too high or too low, we can track down what is the problem, if it's a reagent, if it's the instrument setup, or whatever else. So we use it now on a regular basis twice a year. So in summary, standardization is an opportunity where we can nail down and eliminate all the differences in instrument setup, markers, reagents, panels, and possibly data analysis. And I think that in the future, we will see more of a Lego block approach where part of the procedures will be standardized and others will be tested and benchmarked and gradually improved. And I even hope that what will be more coming in the next years is a possibility to produce a custom reagent sets on demand, so that there will be better stability in terms of dried cocktails, and still a relative freedom in the decision what needs to be in that particular cocktail. I think it's important that we still keep alive the HLDA principles of the CD nomenclature, which is the only customer or wet test type of measurements that assigns the different reactivity of the different antibodies into different CD cluster of differentiations. So we should not let it go, and we should probably think about ways how to make this possible also for the intracellular targets. We should require a complete information from vendors, they are probably in some ways waiting for us to ask for it. Sometimes that makes more complications for them because people will misunderstand and whatever. But I think it's a good practice to go this direction. I think that testing is believing, so we should check or validate your reagents, especially if it's something that you did yourself or where you need to have an increased confidence, and it's not as easy as to just measure CD3 and C. And be aware that the test conditions can be crucial. So with that, I would like to thank to the Euroflow group on the left and to our group at the Child Leukemia Investigation Prague, and I would be happy to accept questions.
Okay, thank you so much, Tomas. That was an absolutely wonderful talk, so we're very much appreciative. We do have some questions that came through, and one of them echoes what I just said, that it was an excellent talk. And they said, thank you very much on behalf of a clinical lab that aspires for more standardization. Do you think that standardization will only ever become reality, at least in the US, if regulatory bodies make it a requirement for accreditation? In your opinion, is that the way forward through accreditation?
Well, you are trying to get me involved in the politics here, I think. But anyway, I believe that standardization, at least the way I see it, is something that should be facilitated, that should be useful. And I'm not sure that the push for it from the, let's say, regulatory authorities is necessarily a good thing, because there is many assays that are not as easy to standardize. And if you say that only things that are standardized will be ever allowed to do in our clinical samples, this will be a big problem. And this is actually where we are in Europe now, because you may not be aware of, but there is a new that was issued in 2017, and it means that by May 2021 in Europe, everybody will be required to measure only CE-IVD assays, and if this is not available, we will have to perform laboratory device test that will be measured against the same set of criteria as if we were manufacturer. So quite tough. And I think it will cause some problems for the kind of assays for small patient groups. So I think to execute some pressure, but I would rather lead by example than by push by force, if that can be my answer.
Okay, thank you, Tomas. One of the questions that that comes through really is, for me at least, is talking about controls and and standardization. I know there are a lot of people, at least in research, but I've heard some comments as well from clinical about reuse of compensation matrices from day to day and going through that process without running the controls on a daily basis. And I'm just curious your thoughts on labs that use those practices, taking into account tandem degradation and potential tandem degradation and instrument drift, and what your personal opinions are there.
So I know that there was a recently debate on a cytometry Purdue list about this. The reality in a clinical labs is definitely to reuse the compensation matrices. I would say that we should see a clinical application of a panel as part of an experiment, and that experiment is not going on for 3 hours, but rather this experiment is going on for maybe a year or half a year. So in that sense, I think that we should make sure that the conditions of everything, the instrument and also the reagents, are unchanged over this period of the experiment, and that makes it possible to reuse the compensation matrix. So that's one group of conditions. And the second one is you have to always test whether your clinical or other assay that you want to use over a prolonged period suffers a significant damage if you account for the instrument drift or antibody degradation. So if your assay is sensitive for those changes, then redesign the assay or don't use it. And if your assay is not affected by small changes in compensation matrix, I would think test and then make your decision whether it's good enough or not. And if it's good enough, I think it's safe to use it. But don't trust it blindly. Don't say because Tomas Kalina said you can use it for 3 months, just do it. Or Euroflow said, just do it. I say always evaluate what you have and evaluate the factors that contribute to possible degradations to your assay. And if you can prove that it does not degrade, that you have a full control of your reagents, then reuse it.
Thank you, Tomas. And then one thing I was curious about is, you know, you showed the comparison across instruments, right? And that you can run the same assay across instruments, which is a wonderful thing. And I'm just curious if you saw any major differences of note or just any observances across PMT-based instruments and APD-based instruments, right? So you showed the Cytoflex, and that's APD-based, and then some of the more traditional PMT-based. Do you have any observations there, or not really?
So I have tested several assays that we brought from Canto to Lyric, which is same type of detection, but more advanced electronics slightly. We took them over to spectral cytometers like Sony, which is PMT based, on Aurora, that is APD based. And I can say this much. So it's a glass that is half full and half empty. So if you want to find arguments that one particular newer instrument is way better than the others, you will definitely find few data points that would show you and clearly show that this is much better. If you want to claim that you can pretty much transfer any assay from one to another, and that's fine, you can also find examples of that. So I don't think there is a simple answer, although we do see differences that less noisy electronics is better in terms of spread, a more precise and more efficient detection of the far red or red dyes on APDs is also better in terms of calculated spread from compensation or deconvolution. So there are improvements for sure, but overall, I would say it's not a different continent. It's just a variant of a similar basis. So in fact, the light is still light, the excitation is still excitation, and we only alter detection. So there are improvements, and you will appreciate them in a high complexity assays for sure. There are if you only want to use a low complexity assays, you will not see that dramatic effect.
Great, thank you. And then one more question for you, just to wrap it up here, is, you know, when talking about, you know, specificity of the antibodies and binding to specific epitopes on the surface of cells, one of the biggest things I think of, or one of the things that I think of at least, is the dissociation of tissue or cells and the impact of certain reagents on those surface epitopes. So just a general curiosity question, do you have any preferred reagents for cell dissociation, or is it very much tissue and cell type dependent?
So I have really no experience in a tissue disaggregation. I do have some experience in a fixation, which sometimes is connected to the dissociation. So the short story is that you are on your own, and there is nothing better than carefully think about your experiment that will tell you what is the impact of your tissue dissociation method on your detection. So for instance, take an easy cell that you understand, maybe peripheral blood, and then subject it to your protocols and read out whether your measurements were comparable to the measurements done without any of these methods, or if they compare well. And step by step, you have to build your knowledge that is particular to your particular to your set of disaggregation and methods. So really, this is very much a field that is, I would say, a perfect opportunity for more standardization, but in the same time very difficult because we still cannot agree on a simple and universally applicable protocols that we can use as a benchmark.
Makes perfect sense. And I think we had another question come through, and I think we have time for just very quickly this one last one. So hi, Tomas, great talk. Using the same target values across the instruments and daily adjustment of the Cytec assay settings is the Aurora's paradigm. So for this full spectrum unit, has the Cytec Aurora been used by Euroflow? And if yes, how do you assess its uniform across instruments?
So we have only started to use the Cytec instruments as of, I believe, 2 years ago in the consortium. So we still gather more experience. Have any answer for you, unfortunately, but you are right that they perfectly well use the concept of fixed voltages and the same as Sony. And I think this is one of the clear indications that this is possible, and we should not be so afraid of those concepts.
Okay, thank you so much, Tomas. We really appreciate a wonderful talk, and yeah, thank you again.
Thank you. Bye-bye.
Yes, thank you, Tomas.